Waxworm saliva is the key needed to sustainably recycle polyethylene plastic. The problem: when plastics were first invented, they were viewed as a wonder material. For many applications, they still are. Today, we benefit from technological progress made possible by them, but we are also just now starting to come to terms with the unintended consequences of them becoming ubiquitous. Microplastics, which are formed when larger pieces are exposed to the environment, can be found on all types of surfaces, both indoors and out. They have been detected in fish, soil, air, lakes, rivers, and even raindrops. It is no surprise, then, to know that they have been found in more than half of human organ systems, and are assumed to be present in most individuals. The majority of our exposure to them likely occurs through indoor air inhalation, food ingestion, and drinking from plastic bottles. The long-term health effects aren't well known yet, but microplastics have already been shown to injure our lungs and digestive systems via oxidative stress. A potential solution: our body's accumulation of synthetic molecules is not good, but that factor is fixed. And due to its incredible utility, completely tossing aside one of our greatest inventions is simply not feasible. The potentially solvable aspect of this issue is that plastics are not yet currently biodegradable. Since there are no species that possess the right molecular machinery necessary to break down their long polymer chains, plastic molecules are unable to complete the carbon cycle, causing them to accumulate in our environment. At least, that is what we thought until 2017, when scientists in Spain discovered that the waxworm's saliva has the ability to degrade polyethylene by chemically breaking apart its backbone. This was precisely the necessary step required for it to re-enter the carbon cycle. Later research identified 4 proteins that contribute to polyethylene degradation, those being Sabelles and Demetra, Ceres, and Cora. While polyethylene is not the only omnipresent plastic, it is approximately 30% of global plastic waste, and fully understanding the saliva's abilities at a molecular level may allow us to find, or even genetically engineer, future degradation proteins that can break down other types of plastic, as well as develop industrial-scale operations that can integrate it back into the carbon cycle. G. melanella, otherwise known as the waxworm or the greater wax moth, is a holometabolous insect, meaning that it undergoes complete metamorphosis from an egg to a larva to a pupa and finally an adult wax moth. As a larva, it feeds on a bee colony's honeycomb as it prepares to pupate. Its ability to consume beeswax is why we believe that they have evolved the 4 aforementioned genes. As far as we are currently aware, it is simply a very fortunate coincidence that polyethylene is chemically similar enough to beeswax to allow the same proteins to break both down. Methods. This semester, as part of Dr. Gordy's Cure Lab course, Genetics 312, our class wanted to know if the Cora gene was conserved across wild and commercially bought populations. We collected DNA samples from several dozen waxworms of both types, then sequenced them, giving us the specific nucleotides of their respective Cora genes. We accomplished this by first extracting the DNA to isolate it from the rest of the cell tissue. Second, using polymerase chain reaction, we amplified the Cora sequence. And finally, we sent the samples off for sequencing, using the Sanger method in order to give us nucleotide-specific data. Results. Most of our class successfully sequenced the Cora gene, for which we corroborated its identity using the BLAST database. Collectively, our data found that the Cora gene was highly conserved among both wild and commercially bought waxworms, and most sequences were greater than 95% identical to the reference sequence in BLAST, indicating that there is very little variation between wild and commercially obtained worms. My particular sample was a commercial worm with 97% identicality. This data also tells us that this gene is likely critical for their survival, or, at the very least, that harmful mutations cause a loss of fitness. Therefore, we can reasonably assume that Cora has a stable role in G. melanella's development. Along with other studies, these results indicate that Cora, along with the others, contribute to the waxworm's ability to degrade polyethylene. Future directions. The high conservation of the Cora gene suggests that Cora is a stable biological tool. Further study of the 4 currently identified proteins is necessary in order to determine the exact contributions of each in this process. These genes may just be the molecular key necessary in order to unlock the final step in the carbon cycle of polyethylene, allowing us to create a blueprint for mass biological recycling on an industrial scale. This would allow us to significantly reduce the amount of polyethylene waste entering our environment, and could lead us to developing similar processes for other common plastics such as polyethylene terephthalate, polypropylene, and polystyrene. Thank you for watching.